Hi everybody, thanks for joining. I might just wait a couple of minutes just to let some people come in and then we'll make a start. Okay, so again, thanks for joining. My name's Tamara Leclercq from the Precision Medicine theme at SAHMRI. And today we're going to hear a couple of presentations on animal imaging capabilities at SAHMRI. First, we'll hear from Wick Lakshantha, who is an imaging scientist and NIF fellow from SAHMRI, and he'll be covering the more small animal imaging that we have. And then secondly, we'll hear from Georgia Williams, who's a research radiographer and also NIF fellow, and Georgia will cover more of the large animal capabilities at PIRL. So again, thanks for joining, and I'll hand over to you, Wick.
I think you all can hear me, and thank you so much, Tam, for introduction and organizing the webinar for us in imaging at SAHMRI. So we are part of a national imaging facility. We spread out Australia. We have 11 nodes with 10 nodes with 17 plus instruments and 34 fellows all around Australia. And we are part of it, SAHMRI, including our University of Adelaide and University of South Australia. And in summary, we have range of modalities, including magnetic resonance imaging, positron emission tomography, and single photon emission tomography, and also X-ray computed tomography and optical imaging. And our animal imaging modalities have two separate section for small and large animal. So the small animal section based in bioresources in North Terrace, and the large animal section based in PIRL at Gills Plain. So we have separate imaging instruments for each animal sections in bioresources and PIRL, as listed in here. I'm going to start with the small animal imaging modalities. In our capabilities, we can cover anatomical imaging, including organs and tissue morphology, and also physiology and functional imaging, which includes the organ and tissue functions, and also the molecular image. Basically, we reach out the molecules, active molecules and cells. So we can cover these sections separately through several instruments available at SAHMRI, including Perkin-Elmer IVIS spectrum, Bruker Albira PET SPECT system, and Bruker MRI system, and a micro-CT, as well as echo MRI. That's a special kind of instrument which uses the magnetic resonance technique to measure the body composition, mainly fat and water content of a tissue or animal. And first of all, I'll start with the molecular imaging. So usually in molecular imaging, we use a target molecule, and we use a signaling component with a target component in a molecule. so that our signaling component emit a signal. It could be a gamma ray or a optical photon, so that we can detect it through a imaging device. So that's the overall picture of molecular image. So in our capabilities, we have three separate instruments which can detect molecular based signal. So PET, SPECT, and IVIS. And PET and SPECT, basically we are looking into a gamma ray emitted by a radioactive isotope. And in IVIS, we use an optical photon coming from a molecule. So I have included two example images here for PET SPECT, as well as the optical image, optical signals coming from animals. So here we are using a molecule or a radioactive element or isotope to find out our target cell. And in the previous one, we split our molecular imaging into two separate sections. One is the nuclear-based method, and the other one is the optical-based method. In nuclear-based method, we have two separate imaging modalities. One is PET. In PET modality, we use isotope which emit positrons. So usually positrons interact with regular electrons in tissue, which prompt a gamma ray as a unique gamma ray of 511 keV, so that we can detect this gamma ray and we can locate the target cell. So basically what we do is we inject a molecule or a nanoparticle or some kind of a drug molecule which contains an isotope or an unstable radioactive isotope, so that we can detect that molecule within the animal. So the other technique is the SPECT. We call it single photon emission computer tomography. In this technique, we use again a radioactive isotope which decays into a stable isotope by emitting a gamma ray. So in this technique, we have a broad range of energy value. We can go from lower from 40 kV to 250 kV. So again, we do the similar experimental setup. We inject the animal with the molecule or agent with radioactive isotopes, and which emits a gamma ray, so that we can detect the gamma ray through a SPECT detector. So this is our Albira system available at Bioresources. So this is a Bruker Albira system which contains SPECT and PET detector. So it's a dedicated PET detector and a SPECT detector within the same system. So in this system, we can detect the gamma ray emitted through the positron annihilation process and the gamma ray emitted by decaying radioactive isotopes. So in this system, usually we have an animal management system and two detectors for SPECT and PET signal. So this is the process. Usually we inject the animal and we can drive the animal through these two detectors to collect our gamma ray signal. So we use the sophisticated Albira software suite, which can detect both SPECT and PET signals and do advanced image reconstruction within a short time period. So then after the next step of this imaging, we have PMOD software toolkit for the further analysis of these PET and SPECT images. So the PMOD software toolkit considered as the gold standard for the PET and SPECT image quantification. So this software toolkit allows us to quantify the PET and SPECT images with great accuracy and do the statistical analysis as well. So if you go to the specification of these two systems, PET system, which comes with the cerium doped lutetium yttrium orthosilicate detector. It's a really advanced detector system, which gives us really good spatial resolution in millimeter range. And also this detector configuration allows us to do depth of interaction corrections for our PET signal, PET gamma ray signal, so that we can obtain no blurring in images. So that's really important when you are doing the localization of the active tissues in animals. So that's a really good advantage with this system. So also this system can have high sensitivity factor up to 9% with the detector ring configuration. And also the system gives us really good count rate and energy resolution, and also higher field of view so that we can accommodate both mice and rats in this system. In SPECT system, we have a dedicated pinhole detector containing cesium iodide single crystal pinhole detector, so that we have high sensitivity count rate up to 2000 counts per megabecquerel. So the system is highly sensitive to low dose image. And also this has higher spatial resolution up to sub-millimeter levels with the high resolution collimator system. So this picture shows the basic configuration of this detector. And also this Bruker SPECT detector system has a broad energy range from 30 to 400 keV. So that allows us to use the vast range of radioisotopes. So that's another advantage of using this Bruker system. And again, this system allows us to accommodate both mice and rats with a large field of view. And here I have shown two examples with the PET system, Bruker PET system, with molecular image. So the molecules are attached with the radioisotope. The first example is using zirconium-89. So this is a really good radioisotopes for longitudinal studies spanning several days, because zirconium-89 has 3 days, 3.3 days of half-life. So we can extend our imaging experiment for several days using this kind of isotopes. And if you compare these two PET and SPECT systems, we can find and list several important advantages and disadvantages. If I start with SPECT system, the main advantage would be the large catalog of traces with longer half-life. That's really important if you are doing longitudinal studies with a long long period of time. And also, we have capability of distinguishing different isotopes using their unique emission energies. So that's another important advantage with SPECT system. But the disadvantage would be we only get 2D images in this system. Then, of course, we can do 3D construction using the 2D images. And also, the other disadvantage would be the semi-quantitative data. We only get semi-quantitative data with SPECT system. And also, it has physical and the technical limits lead us to low sensitivity and limited resolution. But if you are going into clinical sections with PET, or we are going to translate these studies to clinical stage, we can find that the widespread applications in human clinical applications with SPECT system. And with the large catalog of traces, it's really easy to use in a lot of clinical studies. So that's the main advantage with SPECT translating into clinical stage. And also, there's another important point I want to mention. In our SPECT system at SAHMRI, we have a dedicated SPECT camera, because most of the small animal SPECT systems comes with modified clinical detectors. So if you have a modified clinical detectors, they have a lot of drawbacks with small animal imaging. But here in SAHMRI, we have a dedicated Bruker SPECT camera, which gives us the edge in our imaging capabilities at SAHMRI. In PET system, the main advantage would be the high sensitivity. We can go up to 100 or 1,000 times sensitivity compared to SPECT system. And also, the PET image acquisition is 3D acquisition. So basically, we are collecting a 3D image or the 3D raw data at the beginning of data acquisition. So that's an advantage with PET if you are considering localizing your signal. And also, PET is a dynamic imaging method, so that we have good temporal and kinetic resolution. But this also comes with a physical limit. If you consider the positron emission, usually positrons travel around the location of emission, so that it can go up to close to 1 to 2 millimeters depending on your isotope. So that gives us a physical limitation of localizing the signal. And the PET is, we can quantify the PET signal. So that's a main advantage with PET imaging. And also, in SAHMRI, we have another advantage because we have an in-house cyclotron. So this allows us to use short-lived isotopes for our imaging. Because with PET imaging, one of the main disadvantages would be the short-lived isotopes. as I listed in disadvantage. But we can rule out that disadvantage with our in-house cycle so that we can use short-lived isotopes such as carbon-11, which has 20 minutes, and nitrogen-13, which has 10 minutes half-life. So those isotopes are used in drug delivery studies and a lot of the molecular-based studies, so that we can rule out this disadvantage with our in-house cyclotron. And also, if you go with the animal and clinical setting of PET imaging, we can translate our PET imaging to the next level with advancing technologies such as high-density avalanche chambers. So that's the emerging the detector system in PET scanners, and also the MITRU-PET technology. And later we'll see that our new PET-CT system, which is capable of doing certain micro-level PET scanning. So that's the bigger advantage if you wanted to extend your PET imaging into next level. And if you wanted to translate the PET imaging routine into clinical stage, it's really advantageous because the FDG is a routine clinical method used in oncology. And also the PET imaging has become common method used in clinical neurology and cardiology with the developing technology and the merging new methods in imaging neurology and cardiology. So that's a really good advantage if you wanted to translate your studies into clinical stage from animal studies. So the next the topic I wanted to cover is the optical imaging, which also attached to the molecular imaging. So why do we need this optical imaging? So this is a really powerful labeling technique because we can find a lot of the molecules that we can label with the luciferase. So that's a really good advantage with optical imaging. And also the amount of light is proportional to the number of active cells. So that this is this method is highly quantitative. So that's a good thing. And also you can find a range of application from oncology, infectious diseases to stem cell development. So also this is a non-invasive method. So that's a good advantage if you are doing a longitudinal studies. And also the experimental setup would be simple and relatively easy to operate. So in optical imaging, we have three separate techniques. The bioluminescence, that means the chemical process emits a photon, and the next would be the fluorescence. So basically we excite the molecule, and then the molecule emits the photon, so we can detect that the emerging photon from the molecule. And the third, the optical imaging method would be the Cherenkov radiation. So basically here we use the radioactive isotope, which emits the positron or electron, so which translate the energy into optical photon, as shown here. So that means we could use the PET imaging isotopes to produce the Cherenkov optical spectrum to produce the optical photon to detect the molecule. But the main disadvantage would be the absorption. So you have really short path length so that your optical photons will be absorbed by the tissue within short distance. So that's the main disadvantage with the Cherenkov optical emission. And in optical emission, we have vast range of possible applications from the luciferase, which can be used to mark different cells, and for fluorescent proteins and fluorescent dyes, including nanodots and nanoparticles, which can be used to label cells, bacteria, and different types of molecules such as proteins. In our optical imaging system is Perkin-Elmer group, Perkin-Elmer IVIS spectrum, which can accommodate all three optical emission regimes: bioluminescence, fluorescence, and Cherenkov imaging. So in our system, we have high sensitive CCD detector with high resolution field of view. And also this contains 28 filters, wavelengths ranging from 419 to 850. So it goes up to the near infrared region. And also this system capable of doing spectral unmixing using discrete bandpass filter. So that means we could use more than one emission molecule so that we can separate them using the bandpass filter. And also this system can do both the Epi and transluminescent fluorescence. So that's another advantage with the system. And also the software toolkit comes with the Perkin-Elmer IVIS can produce 3D optical tomographic reconstruction using both bioluminescence and fluorescence. And that means we have vast range of applications with IVIS spectrum, from infectious diseases, neurology, inflammation, and oncology. And the main advantage would be, that's what I wanted to emphasize here, we have high sensitive optical detection system so that it can go up to a few cells. So this is one example I have shown here. So we are looking at 3 cells in here. So that means we can quantify the number of photons, and then we can relate this the number of photons to number of cells. So this is highly quantitative method when it comes to the small animal imaging. So that tells us the disadvantage with this system. So this is really sensitive to the path length. So the optical absorption, I have shown here the optical transmission through 1 centimeter tissue. You can see here the lower the wavelength has really low relative transmission. So that tells us this is really sensitive to the tissue thickness. So that's the biggest advantage with the IVIS system, so that we only can do short range observation. So that's the biggest advantage when you are going into clinical studies. So still, we don't have any clinical applications with optical imaging. So the next one would be the MRI system. We have Bruker 1T MRI system. So we can do anatomical scans with small animals. We can accommodate both mice and rats. So this is a desktop MRI system with permanent magnet. So this is capable of doing small animal imaging with a good tissue contrast, and we can obtain good anatomical features with higher a lower signal to noise ratio. That would be the biggest disadvantage with low field MRI system. So if you check out these example images I have shown here, you can see higher lower signal to noise ratio comes with this system. But again, this system can do certain degree of functional imaging too with the appropriate contrast agents such as the paramagnetic nanoparticles. I have shown one example here with manganese dioxide. So it is capable of doing some functional imaging with proper contrast agents. And with this MRI system, we can use this system to cross-platform imaging with our Albira PET system. So, so that we can co-register our images with with MRI as well as the Albira system, so that we can use our PMOD software toolkits to fuse images with anatomical and functional data. So that's a really good advantage if you want to do the functional imaging as well as anatomical imaging. As shown here, two examples with sodium fluoride and gallium-68 imaging with PET and MRI system. So I have few more examples with PET, MRI PET co-registration, so that we can easily localize our functional details with anatomical details, so that you can obtain more localized information in your images. And the next modality would be the MITRU CT system. We have Bruker Sky Scanner. So this system has a minimum of 9 micrometer pixel size, and also we have realistic image visualization with the software toolkit, Bruker software toolkit, as shown in here. And also this system has ultra low dose scanning regime, which gives us advantage in longitudinal studies with minimal radiation effect. And also this system comes with the physiological monitoring system for breathing and chest movement. And if you compare our small animal imaging techniques, we can summarize as in this table. So we have micro CT, MRI, PET SPECT, and optical system, and they have a resolution as shown here. We already discussed those details, and also this this modalities gives us use of probes for functional imaging and obtaining functional information too. And also I have listed some of the drawbacks which we already discussed. And then I just want to quickly mention about our the cyclotron facility at MITRU, so that we can produce various kind of radioisotopes with molecules at MITRU. And also we have a NIF chemist, radiochemist fellow at MITRU, where we can do all sort of chemical or molecular production at MITRU, which accommodate G cyclotron with the various the isotope production capabilities. And also we have another extended facility with NIF network. We are preparing a mobile app with Radiotracer Database, so that you can find your radioisotope throughout the NIF network, National Imaging Facility Network. So far we have 133 tracers throughout our NIF network. So through this mobile app, you can find your molecule, and you can contact the producers, and then you can design your own experiment. So that would be really good advantage with our NIF network. So I'm gonna end my small animal part here. Then I would like to invite Georgia to continue with large animal imaging at PIRL. Georgia.
Thank you. Hi everyone, I'm Georgia. I'm based out at PIRL. I'm just going to give you a brief overview of the large animal imaging facilities we have here. So to start off with a little bit about PIRL, is we are a SAHMRI satellite site located in Gillies Plains, which is about 20 minutes northeast of the city. We have 5 surgical theatres, designated post-mortem room, loads of imaging modalities, and 25 acres of paddocks for large animal holding. It's kind of a bit like a farm. We also have various accreditations, including PC2 ability to conduct research, GLP, and some other ones listed there. So we can also source and order heaps of different animal species, including sheep, pigs, rabbits, guinea pigs, rodents, alpacas, even ferrets. So a little bit about the imaging modalities that we have here. We have an MRI scanner, CT, fluoroscopy, portable radiography. We have CT PET currently being installed. And we also have a small animal lung imaging capability. Most of our imaging machines are human systems that we're using for large animal research. To start off with, our flagship instrument is the Siemens Skyra 3T. So it's a 3 Tesla system, so a high field unit on the higher end of clinical magnetic field strength. It's a Siemens unit, so we have a research collaboration with Siemens, which means we can test works in progress sequences. So you can see here this colourful looping image is a 4D flow sequence from a pig. So we're essentially being able to quantify the flow without using any contrast medium. So we can do various different protocols encompassing neuroimaging, spinal cord imaging, cardiac imaging. We can do angiography without contrast, MRA. We can do spectroscopy, and we have a lot of different coils that we can use. We can also perform spectroscopy to measure biochemical markers in the brain, for example, choline, lactate, etc. So we also have a CT scanner, so we can do various different studies using this unit, including orthopedic work, neuroimaging, forensic imaging, and we can make nice little reconstructions. Oh, skipped one. So our 4D Medical Permetium is a small animal imaging unit. Essentially, it's driven by 4D Medical XV technology, which is a biomedical company in the US and Australia, and we have a collaboration with them. There's only a couple of these machines in the world, and it's essentially the first commercially available dedicated preclinical imaging system. So it can accommodate small rodents up to the size of a ferret. And then 4D Medical can do analyses on the imaging, as you can see in these color maps here. So it can be suitable for various different lung imaging studies, including studies of asthma, COPD, cystic fibrosis, lung cancer, and I suppose ARDS and topical areas of research at the moment with coronavirus. So this is just a little graphic here showing some of the imaging analysis performed by 4D Medical. So you can see they've got colourful maps where they quantify the ventilation. So you can assess ventilation and perfusion using this technology. So now to move on to our X-ray capabilities. So we have a multi-purpose cath lab, which was installed earlier in the year. So this is can be used for an array of applications, not limited to cardiac work. So we can also do vascular studies, respiratory studies, interventional procedures, basically anything that you might like fluoroscopy for. We also have portable intraoperative imaging units, or otherwise known as C-arms or image intensifiers. So we can take those into our theatres and localise devices and anatomy, and also assess vessel patency through administering a contrast agent and performing digital subtraction angiography. So now I'd like to move on to our exciting new addition, which is being installed at the moment and will be commissioned early next year. So we've got a large animal PET CT unit, Siemens biograph. So you can see here that we can use radiotracers and produce images fused with CT. So that's very exciting, and it's something that we're looking forward to start using. So that's just a little bit about the large animal capabilities here. If you'd like more information, feel free to contact me or Wick. We've got our email addresses listed here. We have a heavily reduced rate available for access for SAHMRI researchers. So we encourage you all to get in touch with us if you're interested in using our services. We would be happy to help you. So I'll just have a look if there are any questions. If there are any questions, you can type them into the QA box up the top. Doesn't look like we've got any questions yet, but if you do have any questions, feel free to contact myself or Wick or Wick. I'll put our emails in the chat box. We'll be happy to hear from you. Thank you for joining us on this Friday. All right. Thank you very much. Enjoy the rest of your day, everyone.
Hi, guys.